Okay, the last part of this section on genetics is translation, which is taking the genetic code that was transcribed into an RNA molecule and translating it into amino acids, which build proteins. So the central dogma was something that was mentioned in the video that we watched, which was a creation of Francis Crick of how the information is able to get from the DNA out to ribosomes to make amino acids. And we know now that it goes by way of mRNA. So this flow of genetic information from the DNA to the RNA to the ribosomes to make the proteins is referred to as gene expression. So when you're talking about the movement of genetic information, we first start with DNA, and the process of DNA making copies is called replication, occurs in the nucleus. This information is everything that our cells need to make us, to make our different characteristics, but again, it's tied up in this double helix, this large molecule. So in order for it to get out to the ribosomes to make proteins, it's first transcribed. It's transcribed in the nucleus into the mRNA, and then that leaves to go to the ribosomes. And then in the ribosomes, that's where this information, this little genetic sentence right here that codes for a protein, is translated into the different amino acids that link together to make the protein itself. So what I have here for you is just a little practice that we'll kind of run through here to show you how this translation works. And just like replication and transcription on paper, it's really easy. So we're looking at this little genetic sentence here on our messenger RNAs. Remember, M is for messenger. And we have AUG, GUA, and UGA. So from here on out, we need to be thinking about our nitrogen bases in sets of threes. Now, each set of three is called a codon, and each codon codes for an amino acid. And I'll show you how that works here in just a little bit. So what we're going to do is we're going to play around with taking our codons on our messenger RNA and matching them up to what's called an anticodon on the transfer RNA, and then we'll take that information and we will translate it into amino acids. So here we go. Here again is our messenger RNA. There's our little snippet of information for a protein. And now we're going to be working with our transfer RNA. Now, transfer RNA looks like this. It kind of looks like a T. And what it carries on one end is called the anticodon, which is a series of nitrogen bases. And on the other end is an amino acid. Now, there are 20 different amino acids. We don't have to know them, but I will show you how to use a chart to figure out what each of these little codons codes for, which how you figure out which amino acid the codons code for. So we're going to start over here with AUG, and we're going to see that AUG is also known as the start codon. So it's like the beginning of a sentence where you would find your capital letter. You know where the sentence starts. When you see AUG, you know where the gene starts. So you know where to start translating. All right, so we're going to use the RNA alphabet. AUCG is all that we have to go with. So we have an A, and A pairs with U, U pairs with A, and then G pairs with C. So CAU is the first tRNA. All right, now we'll move on to the next one. So we've got G going with CAU. So here's our second anticodon. And then our last one, this is good practice for our RNA base pairs rather, A, C, U. So here is our tRNA, and then now what we're going to do is we're going to use these tRNA anticodons to translate this information right here into protein. And what we'll use to help us translate, we're going to use one of these little charts. So we're actually going to use the chart that's in your textbook, but there's a lot of different charts. And we'll use this little chart to figure out which amino acid that we're dealing with. Okay, this is like the chart that's in your book. It's circular. This is also like the example that they have on the practice questions, the technology enhanced practice questions that they have available on the Virginia Department of Education for the biology SOL. So when you look at this chart, we can see it's a big old circle, but it's actually divided up into 4 parts. And each of those 4 parts starts out with a really large letter. These large letters here in the center represent the first nitrogen base in your codons or anticodons. It depends on what are you dealing with, which one are you working with. Then we get to another little ring here of letters. They're a little bit smaller. These are your second letters, your second bases. And then this little tiny circle out here are your third bases. And then lying out from that, there's a bunch of words and big long words. So these words like leucine and proline and histidine, these are the amino acids. Now, amino acids again are building blocks of proteins. They're the monomers of proteins. There are 20 different amino acids, and some proteins can be 1,000 amino acids in length, so some proteins can be very large. Now, if we take AUCG and we put it together in a sequence of 3 bases, you're going to end up with actually 64 different codons or 64 different code words. So you're going to see that some of the amino acids actually have more than one code word. So if I'm looking here, let's say alanine, alanine is GCG. It's also GCA, GCC, and GCU. So this chart is pretty easy to work. I just showed you start here in the middle, first base, second base, third base, and then there's your amino acid. Now, I mentioned earlier there's a start codon. That start codon, AUG, is actually methionine. So if there's a start, then there also has to be a stop. So this would be your capital letter, your beginning of your sentence, and then the stop codons are your periods at the end of the sentence. So here we have UAA is a stop codon. UAG is also a stop codon. And UGA is the third stop codon. So this is where the gene stops. This is where translation stops. So it begins here and it ends up there. All right, so to practice around with this a little bit, again, this chart that you have there that we use, the circular one, is the one from your textbook. CAU would be the start codon. So this is what we're looking at here. Your AUG, your GUA, your UGA. This is what I'm using as my messenger RNA. That's what I'm going to be using on my chart. So AUG, again, is the start. GUA is going to be valine. And then UGA is one of the stops. Now, if you notice, instead of writing out methionine, valine, I just put METVAL, and that's generally accepted when it comes to writing your amino acids. Now, what's going to hold these guys together are bonds, not duct tape or Gorilla Glue, but bonds, and these bonds are peptide bonds. So a lot of times you might hear proteins referred to as polypeptides or a polypeptide chain. The last thing I just want to kind of show you is the way you can do this pretty easy by yourself. If I have DNA or my RNA, I just want to go ahead and start thinking about it in sets of threes, sets of threes. And each of these set of threes, again, is a codon. And it's the mRNA that I want to use in that little genetic code chart. And then from this, again, AUG, that's the start codon, methionine, and UAA is the stop. So there's like the smallest protein ever. For some practice, we could take each of these and go through them. So we've got TAC in DNA would transcribe into AUG. CAT would be GUA. TAG, AUC. ACC, UGG, UG. And then ATC is UAG. Now, again, you could use an umpteen zillion different examples of these charts. You just Google genetic code chart, you Google amino acid, you Google translation, and you're going to get a bunch of these little charts. But I like to use the one from the book, the little circle one. So you can play around with this on your own and see what you can do with this. And then when you come back to this and you click on it, the answers will be there. All right, see what you can do with this.